So let's talk about the first biomolecule, that is carbohydrates. That is hydrates. अगर मुझे simply chemistry की term में उसको define करना है, then I would just call it as a hydrates of carbon. यानी कि one carbon molecule attached to one water molecule. The ratio of carbon is to water will always be 1:1. So that's my hydrates of carbon, or in chemistry term, we define it as a polyhydroxy aldoses or aldehyde sugar or ketosis. Polyhydroxy aldoses or ketosis, यानी कि functional group will be either aldehyde or ketone in case of this molecule. Carbohydrates, which we broadly classify into three categories, that is what we call it as monosaccharide, where the term saccharide यानी कि sugar, oligosaccharide and polysaccharides. Okay, so when I say monosaccharide, these are the simple sugar which cannot be hydrolyzed further. I repeat, monosaccharides are the simple sugars which cannot be hydrolyzed further. Whereas oligo का मतलब होता है few, यानी कि oligosaccharide are the molecule which on hydrolysis give rise to 2 to 9 monosaccharide units. Okay, यानी कि ओलिगोसैकेराइड जो छोटी सी शॉर्ट चेन होती है वो 2 से 9 मोनोसैकेराइड यूनिट्स की बनी हुई होती है, whereas पॉलीसैकेराइड, poly many, made up of more than 10 monosaccharide units. Polysaccharides are those molecules which are made up of more than at least 10 or more. It can be 6,000, 30,000 or even 2 lakh molecules of monosaccharide units. So we will be discussing each of this in detail, starting with first monosaccharides. Look at the classification of carbohydrates. As I said earlier, simple sugars, that is basically monosaccharides or oligosaccharides, and the macromolecule, these are my polysaccharides. Monosaccharides are small molecules which are sweet in taste, radially soluble in water, crystalline structures, and name always ends with OSE, O's, glucose, sucrose, raffinose, stachyose. So these are all my simple sugar molecule which are classified further as mono or the oligo. हम most commonly oligo का जो form देखते हैं, that is what we observe as disaccharide. But there are definitely trisaccharide, tetrasaccharide, pentasaccharides as well. But हम अभी के हमारे discussion में disaccharide तक ही discussion में जाएंगे. But yes, when I say monosaccharide, a molecule which cannot be further hydrolyzed, and it is represented by the formula CH2ON, or you can say CnH2nON. This is going to be my generalized formula for monosaccharides, जो हम minimum 3 carbon monosaccharides से 7 carbon monosaccharides तक molecule देखेंगे. When I say disaccharide, यानी कि 2 monosaccharide unit का बना हुआ, trisaccharide 3 monosaccharide unit का बना हुआ. So there are 2 to 9 monosaccharide units joined to form oligosaccharide, and polysaccharide, as I said, made up of multiple molecules of monosaccharides. And when I say this disaccharide or polysaccharide, 2 monosaccharide units are joined together by glycosidic bond. 2 monosaccharide units are joined together by glycosidic bonds. So when I look at the monosaccharide further, which is further classified as simple monosaccharide, which is made up of CH and O only with the formula CnH2nON, and apart from that there are derived monosaccharide as well, जिसमें कार्बन, हाइड्रोजन और ऑक्सीजन के अलावा एडिशनल फंक्शनल ग्रुप भी होते हैं. So let's see first simple monosaccharides. Look at So now when I talk about simple monosaccharides are further classified based on the number of carbon atoms present into triose sugar, tetrose sugar, pentose sugar, hexose and heptoses. आने वाले चैप्टर्स जैसे कि हमारे प्लांट फिजियोलॉजी या ह्यूमन फिजियोलॉजी में जब हम डिस्कशन करेंगे, ये एग्जांपल्स हम बार बार दिखने वाले हैं. नॉट जस्ट देयर, Even when we talk about morphology and anatomy of plants, you will come across this terminologies more and more. So don't try to remember or ratify all these terms or examples. Just try to understand what is their structure और उसकी composition के basis पे हम उसको कहाँ यूज करते हैं फिजियोलॉजिकल फंक्शन में लिविंग ऑर्गेनिज्म में, वो बाय डिफॉल्ट आपके चैप्टर्स में कवर हो जाता है. So starting with, for example, मेरा triose sugar, a sugar molecule with 3 carbons. So obviously formula would be C3H6O3 as my simple monosaccharide. Formula goes as CnH2nOn. So obviously the formula of triose sugar is C3H6O3 and the examples are dihydroxyacetone जिसको आप DHAP धाप करके भी बोलते हो, and glyceraldehyde 3-phosphate और phosphoglyceraldehyde जिसको हम PGA भी बोलते हैं. You will find this molecules during the discussion of glycolysis, right? Glycolysis के जो pathway होती है जहां पे 6 carbon glucose is broken down into 2 molecules of 3 carbon pyruvate. One of the substrate you will come across will be DHAP and PGA, that is dihydroxyacetone, which is my ketone sugar, and glyceraldehyde or phosphoglyceraldehyde, that is my aldehyde sugar. Whereas tetrose में जो example है erythrose and erythrulose, you will find them in the pigments in plants, in flowers. So our pigments like anthocyanin, their basic composition you will come across as erythrose and erythrulose. Similarly, pentose sugar like xylose, you will find it again in case of photosynthesis में जब हम Kelvin cycle की बात करेंगे, you will come across this. Deoxyribose को मैं simple में नहीं रखूंगा, we will take it in derived sugar. But apart from that, arabinose, ribose, ribulose. If you can recollect, the most abundant organic protein or most abundant protein in the whole biosphere we talk about is rubisco, right? और अगर rubisco का ये जो RU है, that is ribulose sugar. Rubisco जो आपका एक एंजाइम है जो आपके फोटोसिंथेसिस में बहुत ही इंपॉर्टेंट रोल प्ले करता है, उसका one of the component of that enzyme is your ribulose sugar. Apart from that, ribose, that's your sugar base in case of RNA. Similarly, अगर हम hexoses की बात करें, definitely we are going to discuss in detail about glucose and fructose and galactose, which is our brain sugar, which you will find in abundance in our nervous tissue. Galactose आपको as it is simple form में, as it is form में नहीं मिलने वाला. Fructose आपको fruit में मिल जाता है. Glucose आपको काफी सारे प्रोडक्ट में मिल जाता है. But galactose is rarely found in a simple form. It is always available in the lactose form, जहां से आपको galactose मिलता है. And this galactose, which is one of the very important component of our neural tissue, or I would say neurolemma. Mannose again you will find as a derivative of breakdown product during the process of digestion or during the breakdown of starch into simpler glucose molecule. Whereas pseudohaptulose, an haptose sugar with the 7 carbon, again it will be a part of our photosynthesis Kelvin cycle. So as I said, ये सारे जो नाम आप देख रहे हो वो आपको प्लांट फिजियोलॉजी में या ह्यूमन फिजियोलॉजी में कहीं ना कहीं कनेक्ट होने हैं. So you don't need to ratify them. It is just you need to understand that these are the molecule structure, that this all examples are of simple monosaccharide having the formula CnH2nOn. Next, we talk about derived sugar. So now when we talk about derived monosaccharide, the monosaccharide molecule which are formed by oxidation, reduction or substitution of simple monosaccharide. I repeat, derived monosaccharides are formed due to oxidation, reduction or substitution of simple monosaccharides. So what we see over here are 4 different types of monosaccharides. Starting with first, we call it as something called as alcohol sugar, right? जहाँ पे carbonyl group gets reduced by hydroxyl group, and the example what we see are the glycerol, mannitol, right? These are the example of alcohol sugar. Next is amino sugar, like glucosamine, galactosamine, यानी कि आपने ग्लूकोज एंड गैलेक्टोज में अमाइनो ग्रुप डाल दिया, ग्लूकोसामाइन गैलेक्टोसामाइन. नेक्स्ट इज डीऑक्सी शुगर, लेट्स से राइबोज में से एक ऑक्सीजन कम कर दिया, डीऑक्सीराइबोज, राइट, व्हिच वी फाइंड इन डीएनए. एंड लास्टली acid sugar, examples such as glucuronic acid, galacturonic acid. Okay, so these are my derived sugar, which you will not find them in simple form. They will always polymerize to form certain structure. For example, यहाँ पे glycerol आपको मिलेगा लिपिड में. Glycerol is one of the key component of lipids, whereas mannitol is a storage form of sugar in algae. Okay, in algae this is a storage form, whereas this you will find in chitin, that's a cell wall of fungi or exoskeleton of insects, or even in cell wall of bacteria, prokaryotic cell, you will find them. Deoxyribose sugar, which you will see in case of DNA. Glucuronic acid, galacturonic acid will become a part of your high hyaluronic acid, a polysaccharide. So these are about derived monosaccharide forming such complex or heteropolysaccharide molecules. More importantly is to understand the structure of this monosaccharide unit, because when you understand the structure of monosaccharide unit, then disaccharide and polysaccharide becomes easy to understand. So let us understand the structure of at least 3 important monosaccharide units, that is ribose, glucose and fructose. So now let us understand the structure of few of the important monosaccharide molecules. For example, when I look at this molecule of triose sugar, that is about glyceraldehyde and dihydroxyacetone. In fact, dihydroxyacetone is the only monosaccharide unit which is symmetrical, because on either side of the functional carbon it has the same group and it has no chiral carbon. Okay, in chemistry we say that the carbon, a functional carbon with 4 different substituents, is called as chiral carbon. So यहाँ पे अगर आप देखोगे दोनों जगह पे सेम ग्रुप अटैच्ड है अल्फा कार्बन को, दैट्स व्हाई दिस डज नॉट हैव एनी chiral carbon, whereas rest of the monosaccharide units you will find whatever is the formula -1 would be a chiral carbon at least in aldehyde sugar. In ketone sugar definitely it varies, so you have to check which carbon does not have which carbon have 4 different group attached to it. And that's how, for example, in ribose, if I check 1 2 3 4, this 4 are my chiral carbon, whereas in ribulose I would say this 1 2 3 are my chiral carbon. Similarly, in glucose and galactose you will see 5 as a chiral carbon, whereas fructose will again have a 4 as a chiral carbon. So at times you may get such question which we have seen in past, but what we need to discuss is about the structure of this molecule, where you can see that ribose, which is my aldehyde sugar, whereas ribulose is a ketonic sugar. Similarly, glucose and galactose are my aldehyde sugar, whereas fructose is in ketonic sugar. Also, it is said that pentose sugar and hexose sugar, these 2 type of monosaccharide units occur in open chain or a straight chain form, as well as in the ring form. In aqueous solution, they also occur in a ring form. How are these rings formed? So when I look at the molecule, let's say this is my straight chain molecule of the glucose, a 6 carbon compound with aldehyde group. It is said that the functional carbon makes the ring with the 5th carbon of the chain. Functional carbon यहाँ पे first makes the ring with the 5th carbon of the chain, and it forms such kind of a pyranose ring, because there are 5 carbon, hexagonal 6 member ring and 5 carbon, so we call it as pyranose ring. We call such rings as pyranose ring. Similarly, if I look at the ribose, another aldehyde sugar, but it is 5 carbon. It is pentose. Glucose, which is my hexose sugar, making pyranose ring. When I look at for the ribose, what I can see, ribose, which is my, again, aldehyde sugar with 5 carbons, right? As I said, functional carbon with the second last carbon of the chain. अब मैं जब functional carbon को second last carbon of the chain को connect कर रहा हूँ, in the ring there will be 4 carbon and 1 oxygen. So it will be a 5-member ring with 4 carbons. So when I have a 5-member ring with 4 carbon, it will be my pentagonal in shape, and this ring will be called as furanose ring. This ring will be called as furanose ring. So this is how we put the numbers of the carbon to the molecule. So this is my ribose, which is C5H10O5. Similar ring will be shown by fructose, but there will be slight modification. Now what change? Fructose is my ketone sugar, right? Just in this slide we saw that fructose is my ketone sugar. अब मैं यहाँ पे जब ketone sugar के ये कार्बन को second last carbon of the chain, so again there will be 4 members of carbon and 1 oxygen. Fructose में भी अगर आप देखोगे 4 carbon and 1 oxygen, so it will make a pentagonal ring. It will form a pentagonal ring with 2 carbons going out of the chain. So numbering from this side, or even if I start from here, it does not affect to the carbon number. So you can, in case of fructose, I repeat, in case of fructose, ring would be furanose. Ideally हम expect करते सर ये तो hexagonal hexoses sugar है, तो 5 कार्बन तो एटलीस्ट रिंग में आएंगे. बट अगर आप फ्रक्टोज को देखोगे, which is ketonic sugar, it will show furanose ring. So if I conclude this part with respect to ring structure of the pentose and hexoses, yes, if it's the 5-membered ring with 4 carbon and 1 oxygen, it will be pentagonal furanose, shape-wise pentagonal furanose ring. And if it is 6-membered ring with 5 carbon and 1 oxygen, it is hexagonal pyranose ring. Now, 2 more important properties what we are going to see over here. One is anomer, and second we will talk about epimer. First, let us understand anomer. So, if you observe, हमने glucose का pyranose ring तो बना लिया, but based on the position of the functional group on the carbon, first carbon, you will see something called as alpha glucose and beta glucose. That means position of this H and OH. If H is on upside, then we call it as a alpha glucose. And if OH is on the upside, we call it as beta glucose. It is very important to understand the alpha and beta glucose difference, because when we will be talking about polysaccharide, we will be using these terminologies and we will understand why cellulose cannot be digested by human body, whereas starch can be. Starch भी glucose का polymer है, cellulose भी glucose का polymer है, but human body cellulose digest नहीं कर सकता, starch कर सकता है. Q will see that, but right now just understand, based on the position of H and OH on the first carbon, in the aqueous form, glucose have a alpha glucose and the beta glucose. So this is what we call it as an anomeric relationship of the glucose molecule. Now when we talk about epimeric relationship, epimers in monosaccharides. Now when I look at the epimers, epimers are basically isomers, right, with just the change in the position of OH and H on one of the carbon. For example, जब हम ग्लूकोज को देखते हैं, ग्लूकोज के थर्ड कार्बन पे H और OH का पोजीशन, you will see in mannose and galactose the same third carbon, right? But now when you compare glucose with mannose at second carbon position position of H and OH are different. Similarly, glucose and galactose at the fourth carbon, position of H and OH are different. So I repeat, glucose, which has third carbon, which is same in both mannose as well as galactose, but second carbon of mannose will show different positioning of H and OH compared to glucose. So we call it as epimer of glucose. Mannose at second carbon epimer, galactose at fourth carbon epimer of glucose. Glucose and fructose are also isomer of each other, but वहां पे functional group ही अलग हो जाता है. C6H12O6 है, but glucose में aldehyde as a functional group, fructose में ketone as a functional group. Whereas in this three, functional group is aldehyde only. It's only the positioning of H and OH on carbon number 2 and 4. 2 in case of mannose and 4 in case of galactose are reversed, and that's why they are called as epimers. So one we talked about enomer, that is positioning of H and OH on first carbon. We say call it as a alpha glucose or beta glucose, whereas mannose and galactose are epimer of glucose. As we move forward, let us have a brief discussion on these four important monosaccharide hexoses, starting with first, that is when I say glucose, which we also call it as a blood sugar, right? Because this is a major respiratory substrate of our body or of any organism, living organism, and that's why we call it as a blood sugar. So it's my common respiratory substrate. Whereas fructose, which we call it as a fruit sugar, all the fruits which are sweet in taste is because of the fructose. So sweetness index of the fructose is considered as 100, and that's my fruit sugar, except grape. Okay, in grape, definitely we see glucose as the sugar. So glucose, which is not just called as grape sugar, blood sugar, we also call it as a grape sugar. Whereas galactose and mannose, which you will rarely find them in a free form. They will always be present in some or the other complex molecule, and as a derivative of that complex molecule, you will obtain galactose or mannose. Galactose, which we call it as a brain sugar, reason being it is found majorly in our nervous tissue. Okay. So the neurulema, the plasma membrane of the nervous tissue, shows presence of galactose. The sphingomyelin, again, a component of sphingomyelin is galactose. Whereas mannose, which is found in hemicellulose, which is a part of wood. Okay. So these are all my hexoses with the common formula C6H12O6, where glucose, galactose, mannose all are aldehyde sugar, whereas fructose is my ketonic sugar. Oligosaccharides. The term oligo, few, saccharide, sugar molecule, meaning a carbohydrate with few sugar molecule or 2 to 9 sugar molecules, we call them as oligosaccharide. In that, three important, that is disaccharide, trisaccharide and tetrasaccharide, which we find them commonly in living organism, out of which our major discussion will revolve around this disaccharide molecule. Disaccharide, yani ki those sugar molecules which are made up of two monosaccharide units. Those sugar molecules which are made up of two monosaccharide units, disaccharide. 3 monosaccharide unit, trisaccharide. 4 monosaccharide units, tetrasaccharide. Please understand the difference between triose and trisaccharide. Triose is my simple monosaccharide containing 3 carbon, whereas trisaccharide, that is, let's say an example, raffinose, is the sugar molecule which is formed by glucose plus galactose plus fructose. These 3 together forms raffinose, whereas stachyose is formed by 2 molecules of galactose plus 1 molecule of fructose and 1 molecule of glucose. So there are total 4 monosaccharide units together forms stachyose, 3 monosaccharide units together forms raffinose, whereas sucrose, maltose, lactose and trehalose are formed by 2 monosaccharide units, out of which we will discuss this 3 a little more in detail. But just to give you a hint about trehalose, this is a disaccharide molecule, यानी कि 2 ग्लूकोज मॉलिक्यूल का बना हुआ है, 2 glucose joined to form a trehalose, and it is found in body fluid, or I would say blood, of insects. You will find trehalose in insects. So trehalose formed by 2 glucose molecule found in body fluid of insects. Let us understand a little more about sucrose, maltose and lactose. So now when I look at the sucrose, maltose and lactose, my most common disaccharide molecule, starting with sucrose, which we also call it as a cane sugar, as the source is sugar cane, or also called as commercial sugar, or the table sugar, right? Sucrose, I am sure in lower grades you must have studied with respect to translocation. Photosynthesis के बाद green part of the leaf से, green part of the plant से जो sugar बना है उसको storage sites, that is seed, fruit या roots तक लेके जाने वाला जो form होता है वो sucrose होता है, because osmotically वो less inactive है. So sucrose, which is made up of 1 molecule of glucose plus 1 molecule of fructose. Okay, and it is said to show alpha 1,2 linkage. Now, what do we mean by this alpha 1,2 linkage? We'll discuss in a little while. Whereas maltose, which we call it as a malt sugar, and maltose, which is made up of 2 molecule of glucose, showing alpha 1,4 linkage, and this maltose you will find during germination. So during germination of seeds, जब starch break होता है, hydrolyze होता है, malt sugar, that is maltose, is obtained. Whereas lactose, which we call it as a milk sugar. So now when I look at this milk sugar, which is made up of 1 molecule of glucose plus 1 molecule of galactose. Definitely, human mother's milk is the richest source of lactose for us. And as we know, it contains galactose, which is our brain sugar, and that's the significance of lactose during early age or during growing period, that mother's milk also helps to provide a source of galactose to the developing infant, which is essential for the development of the brain. And this shows beta 1,4 linkage. Apart from that, we also add that both the sugar, that is maltose and lactose, are reducing sugars, whereas sucrose is the non-reducing sugar. Okay. Now, what do we mean by reducing sugar? What do we mean by non-reducing sugar? And what is its significance? We'll understand that. But before that, let us first understand this linkage part. And that helps us to understand the fundamentals of reducing and non-reducing. So now as we discuss sucrose, which we said that made up of 1 molecule of glucose and 1 molecule of fructose, if you look at the ring form of the glucose, which shows pyranose ring, right? Whereas fructose, which shows furanose ring, okay. So here, between the first molecule of glucose and the second molecule or second carbon of fructose, there will be a formation of a glycosidic bond. So over here, there will be dehydration synthesis. There will be release of water molecules. So the reaction, we call it as a dehydration synthesis. And this leads to formation of glycosidic bond. And such type of linkage is what we call it as a alpha 1,2 linkage. And this is basically my glycosidic bond. Now, if you observe over here, both this glucose and fructose, their functional carbon is getting involved in glycosidic bond. So they do not have a free functional group, and that's why they are classified or they are called as non-reducing. Okay. Now, when I look at the maltose and lactose, which are called as reducing, let us understand that. Look at the structure of maltose, which is made up of two molecules of glucose exhibiting alpha 1,4 linkage. So let's say this is my one glucose molecule. Second glucose molecule, and between the first and the fourth carbon of this glucose molecule, there will be formation of glycosidic bond. So let's say this 1, 2, 3, 4, 5, and 6. Similarly, 1, 2, 3, 4, 5, and 6. So here you can see that is between first and fourth. So this is alpha 1,4 glycosidic linkage. Okay. Similarly, when I look at the lactose, which is made up of one molecule of glucose and one molecule of galactose. Over here, we are going to see beta glucose molecule. If you recollect from that anomer discussion, we had said that beta glucose is the one where the OH is on the upside of the C1, right? So this is my beta glucose. Whereas galactose, which is the epimer of glucose at fourth carbon, यानी कि अगर मैं galactose को देखता हूं जिसका ये fourth carbon OH will be up. So here again between C1 and C4, that is between the first and the fourth carbon, there will be formation of glycosidic bond due to dehydration synthesis. So this we call it as a beta 1,4 linkage. Now if you observe in both this case, in maltose as well as in lactose, one of the molecule functional group is available for the reduction, and that's why maltose as well as lactose are called as reducing sugar. So when I talk more on this reducing and non-reducing sugar part, look at reducing sugar are basically those sugar which has a free aldehyde or ketone group, right? Reducing sugar are those sugar which has a free aldehyde or ketone group, which we see in all the monosaccharides as well as in disaccharides such as maltose and lactose. Whereas non-reducing does not have a free aldehyde or ketone group, which we have seen in sugar. So reducing sugar are those which are able to reduce a cupric ion of the Benedict solution or a Felix solution to cuprous. So the color of the solution turns from blue to reddish if in a given solution reducing sugar is present, whereas non-reducing does not have the free group, so cupric ion does not get reduced. So definitely reducing sugar are all monosaccharide, because all monosaccharide functional or a free aldehyde or ketone group is available. Whereas even in disaccharide, maltose, lactose, we could see the availability of free aldehyde or ketone group. But in case of sucrose or even in trehalose, जहां पे हमें alpha 1,1 linkage दिखता है, sucrose and trehalose are both the disaccharide molecule which are non-reducing. Obviously, question है सर हमें ये reducing non-reducing का concept का practicality क्या है? To find out the presence of sugar, or I would say glucose, in a given sample of urine or a given solution, this Benedict test or Felic test is used. So ideally ग्लाइकोसयूरिया के केसेस में यूरिन सैंपल में ग्लूकोज है या नहीं है वो हमें इस प्रॉपर्टी के थ्रू पता चल सकता है. So that is with respect to the concept of reducing and non-reducing sugar. I again repeat, all my monosaccharides are reducing sugar. Disaccharides are reducing except this two, that is sucrose and trehalose. Apart from that, polysaccharide. Now polysaccharide जिसका हम डिस्कशन करेंगे, which are the long chain molecule, may be made up of 6,000, 30,000, 60,000, 6 lakh or 2 lakh glucose molecule. Those are non-reducing sugar. But polysaccharide has something called as right end as the reducing end and left end as the non-reducing end. Why so? Let's have a discussion on polysaccharides. So polysaccharide, poly, many, saccharide, sugar molecule. So now when we have a sugar molecule with more than 10 monosaccharide units, we call them as polysaccharide. Now when I say homopolysaccharide, yani ki those polysaccharide which are made up of same monosaccharide unit. So those polysaccharide which are made up of condensation of same monosaccharide units, we call them as homopolysaccharide. When I say hetero, that means a different monosaccharide units or some prosthetic group are found to be present. They are called as heteropolysaccharide. So when I look at the examples of homopolysaccharide, starting with starch, glycogen, cellulose, okay, all these three are polymer or formed by the condensation of glucose molecules, so we also call them as glycans. Whereas inulin, which we call it as a fructan, because it is formed by the condensation of fructose, and apart from that even chitin or chitin, which is formed by polymerization of N-acetyl glucosamine. So these are my homopolysaccharides. So when I look at the examples of heteropolysaccharide, we take an example of hyaluronic acid, heparin, or even agar medium, which we use for a bacterial growth or a bacterial laboratory research methods, and even chondroitin, which is found in the matrix of the cartilage. So these are my heteropolysaccharide. Out of all this, we will be having a detailed discussion with the structure on this three, whereas rest of the molecules we will have a brief discussion as well. So let us discuss each of this homopolysaccharide or a glycan in detail, starting with first starch. Starch, which we call it as a storage form of sugar in plants, right? Storage form in plants. So when I look at the starch, which has two components, the 20% of the starch is in the water soluble form, that is what we call it as amylose, and the 70 to 80% of the starch is in the form of amylopectin, that forms 70 to 80% of the starch. So let's take an example, potato, one of the very rich source of starch. Potato में 20% will be in the amylose form and 80% in the amylopectin form. So अगर मैं इसमें difference देखता हूँ, amylose form, which is unbranched helical chain and made up of about 200 to 2000 monosaccharide units, that is glucose units, water soluble. Whereas amylopectin, which is branched and it is linear chain. It shows branching and it shows linear chain made up of about 1000 to up to 2 lakh monosaccharide unit, and it is insoluble in water. तो हम लोग जो प्लांट सेल के अंदर स्टार्च ग्रेन्यूल्स देखते हैं, वो ग्रेन्यूल्स का शेप इन अमाइलोज और अमाइलोपेक्टिन के कंटेंट के ऊपर डिपेंड करता है. When I said amylose is a helical structure, so yes, it looks somewhat like this, unbranched helical chain of monosaccharide where glucose shows alpha 1,4 linkage. Okay, and when I say amylopectin, a linear chain with branching pattern, a linear chain with branching pattern. तो जो linear form है, यहाँ आपको अल्फा 1,4 लिंकेज मिलेगा और ये जो ब्रांचिंग पॉइंट है यहाँ अल्फा 1,6 लिंकेज, बिकॉज यहाँ पे ब्रांचिंग पे दिस ग्लूकोज कनेक्टेड टू दिस ब्रांचिंग पॉइंट एट फर्स्ट, सो इसका 6, इसका फर्स्ट, देन अगेन इन द सेम ऑर्डर it will show alpha 1,6 linkage. So that is my starch. Now when we look at the glycogen, which we also call it as an animal starch. The reason I call it as an animal starch, it is storage form of polysaccharide or a storage form of carbohydrate in animal liver and skeletal muscle, right? हमारे skeletal muscle और liver में excess of glucose or excess of carbohydrate is stored in the form of glycogen. So when I look at the molecule of glycogen, which is said to be made up of about 30,000 or as large as 6 lakh monosaccharide units or 6 lakh units of glucose, right? In fact, glycogen का जब हम structure देखते हैं, वो अमाइलोज और अमाइलोपेक्टिन का कॉम्बिनेशन है, यानी कि ग्लाइकोजन इज नॉट जस्ट द हेलिकल स्ट्रक्चर बट इट इज ब्रांच एज वेल. सो यहाँ पे जब हेलिकल स्ट्रक्चर की बात करते हैं वहां पे वो अल्फा 1,4 लिंकेज शो करेगा और ब्रांचिंग पॉइंट्स पे अल्फा 1,6 लिंकेज शो करेगा. In fact, glycogen is more complex and more branched than amylopectin. So when I look at this structure, as I said, it is helical, and this helical structure is also showing branching pattern. So it is helical with branch. So it's like amylose and amylopectin together. Next, we talk about cellulose. Cellulose. As we say, cellulose is the most abundant organic molecule. I repeat, it's the most abundant organic molecule present on the whole biosphere. Okay, as we know, cellulose, which forms cell wall in plant cells, right? When I look at the structure of cellulose, which is linear, unbranched. It is linear and unbranched, and more importantly, it shows beta 1,4 linkage. It exhibits beta 1,4 linkage. अब तक हमने सब जगह पे अल्फा ग्लूकोज लिया था, यहाँ पे हम बात कर रहे हैं बीटा 1,4 linkage. And cellulose threads always occur in bundle. You will always find the bundle of cellulose fibers. So in between the cellulose thread, there will also be a presence of intermolecular hydrogen bond. Yani ki you will see such linear unbranched cellulose fiber with intermolecular हाइड्रोजन बॉन्ड। और जब मैं ये एक सेल्यूलोज फाइबर को देखता हूं which shows beta 1,4 linkage where adjacent glucose are at 180 degree to each other. That means cellulose का जो structural arrangement है, as I said, it's a beta glucose. So yes, OH on the upside, H on the lower side, connected to next glucose molecule, which is placed at 180 degree to the first one or to the adjacent one. So यहाँ पे जो fourth carbon है उसका OH, and here there will be formation of glycosidic bond. Now next glucose molecule will be placed like this. So the first, fourth, there will be formation of glycosidic bond. This is how a cellulose exhibits beta 1,4 linkage, and that's the reason human stomach, or I would say human digestive tract, cannot break cellulose, cannot digest cellulose. The reason is हमारे बॉडी में जो अमाइलेज रिलीज होता है वो अल्फा लिंकेज को ब्रेक कर सकता है, इसीलिए ह्यूमन डाइजेस्टिव ट्रैक्ट कैन डाइजेस्ट स्टार्च और ग्लाइकोजन which shows alpha 1,4 linkage or alpha 1,6 linkage, but human digestive system में जो अमाइलेज रिलीज होता है वो बीटा बॉन्ड को, बीटा 1,4 लिंकेज को डाइजेस्ट नहीं कर सकता, एंड दैट्स द रीजन सेल्यूलोज बिकम्स अ बल्क ऑफ आवर stool, or I would say a bulk of our, I mean, it is obtained as a roughage for a better dietary movement. There are more pointers to cellulose, like cellulose, which is present in cotton. In fact, cotton contains 90% cellulose. So definitely textile industry का जो major component है cotton, और उसमें जो होता है वो cellulose. Apart from that, अगर मैं wood की बात करूं, in fact, the quality of wood will be determined by the percentage of cellulose. Okay, so in wood 25 to 50% is cellulose. Apart from that, cellulose को various chemicals के साथ treat करके जो हम product create करते हैं, that is cellulose acetate, which is again used for forming a wrinkle-free cloth. और अगर आप सेल्यूलोज नाइट्रेट को अगर हम देखें, then this is my propellant explosive. Okay, even cellulose xanthate, which is used in rayon and cellophane tubes, or even carboxy methyl cellulase, which is a very much important emulsifier. Used in cosmetics or even in ice cream industry. So there are n number of activities or industrial activities which are associated with cellulose. I repeat, which is most abundant organic molecule in whole biosphere. So now as we summarize this homopolysaccharide, that is glycan, as we see again starch, where we can see there is a presence of amylose, which is unbranched helical structure, and amylopectin, which is linear and branched. Okay, so over here you will see only alpha 1,4 linkage, whereas over here you will see alpha 1,4 as well as at branching point alpha 1,6. Whereas glycogen, as I said, storage form of sugar in muscles and liver. So it is helical as well as branched. It is extensively branched helical molecule. Whereas cellulose, which occurs in the bundle, so cellulose threads which occurs in the bundle, and these are unbranched, I repeat, unbranched, and exhibiting beta 1,4 linkage, where adjacent glucose molecules are present at 180 degree to each other. Now, what is the significance of understanding whether it is helical or linear? Because if a molecule, if a given polysaccharide molecule is helical in nature, it will hold iodine and give the iodine test positive. दैट मीन्स अभी तक हमने जो भी स्ट्रक्चर स्टडी किए, डेफिनेटली amylose will hold the iodine and will give blue color when it is added to the iodine solution, whereas amylopectin, which is linear, will not be able to hold, but still it will give red color when it is given to the iodine solution. Similarly with glycogen, having the helical nature, can hold the iodine, whereas cellulose, linear structure, cannot hold the iodine. It cannot hold iodine. So these are the MCQ point based on your NCERT. Helical chain है, iodine hold कर पाएगा. Linear chain है, iodine hold नहीं कर पाएगा. Now let us talk about two more homopolysaccharide, very important homopolysaccharides. One is chitin, or what you also pronounce it as chitin, and second inulin. It is not insulin. Do not make an error. Insulin is your protein. It's a hormone. Inulin. So we will be talking on this two more homopolysaccharides, starting with first chitin or a chitin, which is the second most abundant organic molecule. First is definitely cellulose. Second most abundant is chitin, which is formed by polymerization or polymer of N-acetyl glucosamine molecule. Okay, so N-acetyl glucosamine molecule के polymerization से बनता है ये chitin, and this is found majorly in cell wall of fungi as well as in the exoskeleton of arthropods. So all the arthropoda, without any exception, exoskeleton is said to be chitinous or chitinous exoskeleton. So that's a polymer of N-acetyl glucosamine. Now when I look at the inulin, जब हम inulin की बात करते हैं, inulin is the polymer of fructose. This is the only fructan you will see, and this is basically a storage form in Compositae family. That is in roots, tuber and rhizomes of dahlia and artichoke. You will find inulin, which is a polymer of fructose. Now inulin का एक medical application भी है. Now inulin cannot be synthesized, or we call it as metabolized by human body. Okay, मतलब ह्यूमन बॉडी ना तो इन्यूलिन बनाता है, ना अगर उसको कंज्यूम किया तो मेटाबोलाइज कर पाएगा. सो वॉट एवर इन्यूलिन यू इंजेक्ट will come out as it is, and that's the reason it is used to check GFR, or what you call it as a kidney function. जितना इन्यूलिन हमने इंजेक्ट किया, कितने टाइम में वो बाहर किडनी उसको फिल्टर आउट कर रहा है, वो हमें इन्यूलिन के यूज से पता चल सकता है. सो दैट्स इट्स एप्लीकेशन. सो दैट इज अबाउट चिटिन एंड इन्यूलिन. We talk about heteropolysaccharide, meaning a polysaccharide molecule which is made up of more than one different type of monosaccharide units. We come across four major heteropolysaccharide for the discussion, starting with the first one, that is high hyaluronic acid. High hyaluronic acid, which you will find in the synovial fluid, that is in our synovial joint, even in our vitreous humor, that is a part of our eye. You will find this gelatinous fluid which maintains the viscosity and provides the frictionless surface, which is made up of N-acetyl glucosamine and glucuronic acid. If you recollect, both these molecules are my derived monosaccharide. This is my amino monosaccharide, right? Whereas this is my acid monosaccharide. Whereas, so yes, as I said, hyaluronic acid found in synovial fluid, even in the vitreous humor of eye, or even in your basal lamina of epithelial tissue or of the basement membrane. So animal tissue में आप इसके बारे में डिस्कस करोगे. Heparin, a very important anticoagulant, right? जो हमारा ब्लड को क्लॉट होने से बचाता है, रोकता है और ब्लड फ्लो ब्लड वेसल में इजीली चलता है. That is also sulfated N-acetyl-glucosamine, that is amino sugar, and glucuronic acid का polymer. Chondrin, which is found in matrix of cartilage to provide the tensile strength to prevent or resist the compression, is N-acetyl-galactosamine or glucuronic acid ka polymer. So this is not that two molecules are making this. It's a polymer of these molecules. Multiple molecules of N-acetyl-glucosamine and glucuronic acid alternatively forms heparin. N-acetyl-galactosamine, glucuronic acid alternate polymerization forms chondrin, and similarly agar, right? The one which is found in the cell wall of red algae. Apart from that, even agar, which we use in laboratory for the culture medium, it is a mucopolysaccharide, that is a protein substance attached to the polysaccharide molecule. So these are my heteropolysaccharides. So here we complete the discussion of carbohydrates. Next, we will talk about proteins, lipids, nucleic acid in videos to come. Stay tuned. Thank you.